Okay, our next speaker is Anne-Lise Borresen-Dale, who is from the Oslo Radium Hospital. And the title of her talk is Exploring the Systems, Pathology, Biology of Breast Cancer. And this is, follows the beginning of this revolution in understanding transcriptional patterns in different diseases. Anne-Lise.
Thank you, Arne. First of all, thank you for inviting me to come to this lovely place, which is a place when I came here and said, if I'm going to retire someday, it should be around here. And give me the opportunity to tell you about some of our research program and how we're trying, and some of our recent data, and how we're trying to capture the data and the cohorts so we can be able to really explore the systems and understand the systems, pathology and biology of breast cancer, to be able to get better treatment to the patients. And we've just merged 3 big hospitals in Oslo, so we are now the Oslo University Hospital with more than 22,000 employees, so we're quite a big enterprise. But that also resulted in the strengthness of the Comprehensive Cancer Center. So, because what we start with, particularly as geneticists, when we have a patient coming in or we have our control cohorts, we capture the patient's own genotype. Looking for inherited mutation, but also, of course, normal genetic variation using SNP and CNVs, and now move that over to arrays. Very important is to have questionnaire data, and for some of our cohorts, and particularly of the prospective women that we are following, we have extensive questionnaire data about exposure. And particularly when it comes to hormones, childbearing, height, weight, HRT, oral contraceptives, and so on. And of course, we take blood samples, and sometimes we have blood samples prior to disease, and some we have at the disease, and some we have both. And we try to do some blood transcriptomics to see if we can use that as early diagnosis and a marker for exposure of disease. And of course, we save serum for being able to do serum markers. We have started to look for some glycan antibodies, but also for circulating microRNAs and circulating DNA from the tumor. And when the tumor arises, we try to, everyone have a mammogram. So everyone in Norway has mammograms regularly, above 50, which is stored centralized. Some of them also have MRS and MRI, so we have a spectrum from some of our experimental, those that go into our experimental trials. And of course, we take the tumor out and have very carefully pathology, make tissue microarray of all our cohorts. And then we have also an HR mass metabolic spectrum of some of our patients, where we put the tumor into this HR mass machine and get the spectrum out of it to capture some of the metabolic profile. We take out the sentinel nodes and make sentinel node characteristics. We take out bone marrow and look for disseminated tumor cells in the bone marrow. We do that also in the blood. And of course, the tumor itself should be fresh frozen. For most of the occasion that we want to, I usually say that I only participate in study which is TGIF, and that is not thanks God it's Friday, but thank God it's frozen. So we try to capture as much mutation we can, doing amplification and deletion, and start to do deep sequencing. Tumor epigenome, both specific genes and genome-wide. And of course, various type of expression profiling at the mRNA level, at the microRNA, and at protein. And protein, of course, traditionally we have the tissue microarray, which we make, as I said, from all our cohorts. But we also do in collaboration with Gordon Mills the reverse phase protein arrays to get a more quantitative measurement of the proteins. And we try to make as many xenografts as possible, because that's hard. It's getting easier with a new type of mice, but we try to have that as many as possible so we can go back and do experiments with the mouse. And we have sort of surrogate longitudinal studies. We have tissue from healthy women, we have from DCIS, from small invasive, and large and more advanced stages. So what we're trying to do is to see how different are these various molecular profiles at the different levels in the different patients at the different stages. And what factors influences these molecular profiles. And do the different profiles tell us different things? And can we combine them into an integrated approach? And do they have some kind of a hyperstructure? And of course, most importantly, do they have clinical relevance? And can we translate this into clinical management? So we started out more than, actually 12 years ago, when I was at Stanford visiting David Botstein, and they were starting out with these arrays, and I had this set of tumors that I was looking for a technology where do multi-northern. So, because when the arrays came around, that was a fantastic opportunity for us to start to do that. And at that time, we had a 9,000 gene on the array, and we have a limited set of tumors, but they were on the same stage and the same treatment of these patients, and we had before and after samples. So we were able to select genes that showed more similar expression between two samples from the same tumor than between any other tumor. And then we identified these 5 subclasses using these, what we call the intrinsic genes, that had these characteristics. And we know that this classification is very robust, it's seen in very many other data sets, and with at different age and different stage, and with different array platforms. And most importantly, the different subgroups have difference in survival. You see here, this was our first cohort on stage 3 and 4. This is a more recent one with a consecutive series, which you see it crosses over, that the luminal B has a very bad prognosis. And of course, people have done this now over and over again, and what you see is these 5 subclasses that are really robust. When you have large sets like this, this is a set from North Carolina with Chuck Peru, you can see that it comes up some additional classes, like this Claudine Low, which has a lot of infiltrated cells and are a specific class of tumors, which we missed in the first one because it probably wasn't there, because we only had the much fewer samples. And you see that has a very bad prognosis. But you can see the prognosis holds in all the cohorts. So this is something biologically meaningful with that. And even if we now have extended the number of genes that we're looking at, these classification holds up. So what do we know at the mRNA level of these classes? We know that the luminal A and the luminal B, that's a continuum. We know that the ER is prognostic, and we can take other genes that correlate with the ER, which makes it more prognostic, and they have some of the characteristics in the luminal A versus the luminal B, which is more proliferative, more often P53 in the luminal B than in the luminal A, that has more often the PIK3CA and the GATA3, and the luminal B has a much worse prognosis. The base-alike is 10 to 15% of the cohort in our population. In American African, it's close to 50. They are highly proliferative. We think they may derive from a distinct cell type. They have a very short survival, although there is a subgroup that seems to respond very well to high-dose chemo. And we know that the BRCA1 carriers, they develop these base-alike tumors. And they have a very high frequency of P53 mutation. When it comes to the ERB2, we call this now an HER2-enriched subgroup, because this exists both on the ER-positive background and on an ER-negative background, with and without P53 mutations. And there are some on a very strong stromal background, and in our cohort, they have very different in survival, and these are not treated for the antibody to the Herceptin, which is antibody to the ERB2. So, and this is also some characteristic what you see within these subtypes, that now this is a centroid correlation to how similar it is to the original cohort that we had. And you can see, typically you see that the base-alike anti-correlated to the luminal A, and the luminal A anti-correlated to all of the others. And then you have all these that has a little bit of phenotype of everyone. So it can be hard to classify exactly what it is. What we know is the true base-alike, or the typical base-alike, and the typical luminal. And then this is another cohort that was recently published, come from this transbig, these are stage 1 and 2, and these are more advanced tumors. So you see you have fewer of these luminal A in this cohort, and many that are a little bit indistinguishable and harder to classify. But I think this is how far you can get with expression when it comes to this classification. Because we know that tumors are very heterogeneous. So what can other type of analysis give us? So this is our model cohort. We call it Oslo 1. And it started in 1995 to 98. So it is the inclusion criteria. So it's a fairly long follow-up on these patients. There are 920 patients in the cohort. They're all stage 1 and 2. And the trial was to draw the bone marrow and see if presence of bone marrow at the time of diagnosis, and then 1 year later and 3 years later could predict for outcome. We had, unfortunately, only 103 patients that we got fresh frozen tumor from at that time. But this has been our model cohort, and of course we have paraffin blocks and all the other. We have bone marrow and blood samples from the rest of the cohort. So from this is what we call the MICMA cohort, where we have done a lot of studies and have data on very, very many platforms that will be sort of our model building cohort. Now we have another study ongoing, which we call the Oslo 2, where the similar design of this one. Now we get everyone fresh frozen, if it's not too small. And we have 650 recruited for the time being. Around 500 have been extracted, so we are starting to do the analysis on. And we're aiming at 2,000 by the end of 2012. So this concerns all the hospitals in the Oslo region. And then it's another one which is ongoing, which I will come back to in the end of my talk, which is this NOVAC, which stands for Norwegian Women and Cancer, which is a population-based study where we have 150,000 women which we follow over time, prospectively. And in that cohort, we have now recruited 200 cases, and we're aiming at 1,500 by the end of 2013. So this is a nationwide study. So to leave the expression to do what can methylation tell us, and will methylation profiling give additional information, and how much of the expression variation can be explained by methylation? And because we started with this, and this was the first we did, was an Illumina Golden Gate with 800 genes, 1,505 selected CPG sites, 2 to 3 per gene on average. And then we ran through this MICMA cohort that we had enough DNA and some to do the methylation profiling on, and some normal tissue. And what we saw when we did unsupervised clustering is that they clustered into 3 main clusters. And they recapitulated a bit the expression, which you would expect, because the expression is also partly driven by the methylation. But there are subgroups within here, and some jump. You can see that you have some basal coming into this cluster 2, and you have some luminal. The luminal actually spread over this methylation pattern. So could this help us in subclassifying those that are difficult to classify? And if you look into survival of these 3 clusters, this is borderline, but there are a clear tendency that there are differences in survival, which are now independent on the expression profiles. So we got on now to do it genome-wide, and we've done it with Mike Wigler's ROMA technology, which seems to capture some of the same. And we have some methylation profiles that are also highly connected to outcome. So we know because we are generating the methylation to try to integrate that as part of all the type of molecular profiles. So a statistician in my lab has tried to make some first attempt now how to integrate the methylation with the expression. And because if you just now first to look for cis correlations. And this is how it looks like when we take this set of 800 genes versus the expression of the same 800 genes. And this is now distributed along the chromosomes, and this looks very messy. And it seems like it is just around 0. So it doesn't really capture any correlation between the methylation and the expression. But when she did the corrected after multiple testing using the false discovery rate, she was left with this. These are genes that are methylated and downregulated. And of course, we had a couple of up here, which we are exploring, which are methylated and upregulated, which are a little counterintuitive if it really this is it cis. So we have to explore that further. But this is what we want to do much more of and integrate not only 2 and 2 level, but more and more levels at the time. So the next we ask the question, or we want to explore is, what is the microRNA looking like in this set of tumors? And can that define different subclasses? And how much do that recapitulate, and what does it add? So again, it's MICMA tumors. Here we had 101 tumors, and we used the Agilent human microarray with more than 700 human microRNAs on that array, and some viral sequences as well. Coupled it also to the expression. First, what we saw is that we could definitely find microRNA that distinguish between the luminal A and the basal, which are a really typical outlier, or 2 types of different breast tumors that you see. And you can see here that some of these microRNAs perfectly separate the luminal versus the basal, so you can use that as a marker for luminal. And you can see here that some are genes at the p-values that come up that are up in basal, and these are the ones that are down in basals. And we have others, like for example, that are correlated to the p53 status, because we have p53 sequence for that cohort, the total p53 sequence for all of the samples. And here you can clearly see that this is a signature for p53 mutant, and this is a signature for p53 wild type. You have some exceptions here, which you may think that they have a p53 mutant. They do not have a mutation. We have gone back, but it could be that this is a shut-off of the pathway. And here you can see again, we find some of the luminal of the basal-like over here, which do not have a p53 mutation. They end over here. And you can see that some of the luminal B also, they're luminally split into two. So this gives additional information. And again, some genes that are up in the mutant p53 and some that are down in the mutant of the microRNA that are down in the mutant p53, or in the mutant p53. And then we did correlations, correlative relationship between microRNAs. Took each microRNA and then correlated them to all the expression profile, and looked for genes that behaved like, microRNA that behaved like this, that they were correlated strongly with a bunch of genes when they were down and then up here. So when this is upregulated, these genes are up, and when this is downregulated, these genes are down. And here you can see this is completely random when it comes to the subtypes. And this is this MIR. When you look for what are these genes that this MIR 150 are involved in, first of all, when we took the MIR itself, it's highly significant when it comes to survival. But also when you look at the network that are in or the go terms that come up on those genes that this MIR are regulating or are involved in regulating it's the immune system. So I think microRNA will definitely give us additional information. What about the protein? As I said, we have used in collaboration with Gordon Mills, we have used his reverse phase protein array. So we have approximately more than 200 validated antibodies, both phosphorylated and non-phosphorylated. They are enriched for proteins in the AKT pathway. So they are biased in that sense. But what we see here is that we can clearly see that there are differences, for example, in the protein level, now in the measurements of the protein level between the different subclass. In the cyclin B1, D1, and E1, you can see that the B in the luminal B has a very high cyclin B1, which is also similar to the basal-like, but in the cyclin E1 is up only in the basal-like, and this is not caused by amplification, but by elevated expression of the cyclin E1. And if you take these proteins together and look for the level of the protein and group them according to the level of these three proteins, you find three different groups, and you can see they are different in survival, which again goes across the classical 5 subtypes. So this is another technology that we are using in collaboration with the Technical University in Trondheim, who are specialists in MR and MRI, and we're using these in vitro system. So we take out a piece of the tumor and put it into this little core here, and then put it into this rotor. And then it starts spinning, so this is high resolution magnetic angle spinning, because it spins under a specific angle. And you turn back a spectrum, and this particular one is a 14 Tesla, and you get spectrum back like this. So you capture the choline, the phosphokylines, the glucose, and the glycine, and the taurine, and some of the some lactates, and some amino acids, and some fatty acids. So you get a spectrum of metabolic, and of course the height of it and the curve under these is a quantitative. So you can now quantify a metabolic profile for each of the tumor as well. And the beauty is that when you put it in here and take it out, this takes approximately 20 minutes. Very few genes are altered, because this is done under a low temperature. It's not freezing, but it is at room temperature, which is running. So it's a short time, so we know that it's a certain set of genes that alters, and particularly there's the very early response genes. They are altered because it's a stress to the tumor, but we can take it out and do expression arrays on that. And when you cluster these tumors, so this is part of this same set of tumors again, but here we took only out the luminal A tumors, which were the biggest one that we had that we can do this on. And clusters, so this cluster are now based on the metabolic profile and the intensity of the, so you can see they fall into 3 very different clusters, and this is only luminal A by expression profile. And these are the typical spectrum that you see. which you can see that they really vary, and some of the metabolites are really significantly different in these three groups of tumors. So when we look at the expression of these genes, first of all, you can see that several significant differences between the groups were seen. For example, in this group 2, which was the middle one, they were much lower in glucose and they were much higher in alanine. And if you look for the genes that characterized this A2 group, they were genes, using the GO terms, they were enriched for genes that were related to cell cycle and DNA repair. Because this is a small set, so we really have to do that, and we're doing that now for the next big Oslo 2 cohort, which will be 2000. So there we really can see how does this correlate, and does this have any clinical impact. These are far too few also to do any survival analysis. So this is our sort of first pilot trial to see it works. We can get a nice profile out, both from the metabolic profile and from the expression profile from the same piece of tumor. So if we move now from the tumor genotype to the tumor genotype, we know that genomic instability is a hallmark of cancer. And we have used various types of arrays, CGH arrays, and deep sequencing. And the question we're asking is, can the underlying defect at the DNA level be captured by studying the genomic architecture? And does this architecture direct the route of progression, or determine if it goes to a luminal route or a basal route, and can it guide in therapy decision? Because we started out some years ago first with a very classical chromosomal CGH, and then we moved to the first array where we used the cDNA arrays. And Anna Bergamaschi was a student in my lab and worked also with John Pollack, so we did this in collaboration in my lab using the Stanford arrays. And what she saw also now these are now the frequency plot and split now into these expression subgroups. You can clearly see that there are different patterns between the luminal A, the luminal B, the ERB2, and the basal-like tumors. And that these are significantly differences in what you see between them in using this type of array. What was interesting at that time was that when we looked for the total number of the genome of gain and loss, so the fraction of the genome that had gain and loss, it was far more gain and loss in the basal-like tumors. But if you looked at the amplitude, the type of or height of the amplification, that was far the highest in the luminal B tumor. So these are two probably very different mechanisms. So then we moved on to other technology, and this was the Roma technology in collaboration with Jim Wigler's group and Jim Hicks, done again on this MICMA sample sets and some other Swedish tumors. And these were the pattern we saw from them. These are now single tumors. The previous one was a frequency plot. And you can see these type of tumors. Now each of these are one tumor, chromosome 1 to the X chromosome. This is hybridized to a male. That's why it looks like it's amplified. So you can see this is very, very different. And when we looked into this seems to have much better prognosis, and these has a poorer prognosis, and these are sort of some intermediate. But this was based on this Roma technology and the Nimblegene platform, and we saw these, and we thought that they reflected different type of mechanisms, that this was possibly translocation. Could this be DNA repair defect, or could this result from telomere maintenance loss? So we needed to go on. We saw that typically luminal A you found both in these, and the basal you found more of this, but also some of these, and you find the luminal B and the ERB2, a little bit of both. But it was enriched in these three groups for these three subtypes. So now a postdoc, and is a very talented student in my lab, and teamed up with a statistician. And they set out to develop an objective estimate of genome-wide architectural distortion, because the Roma algorithm only worked on that particular platform and with that type of segmentations that you did. So they really wanted to do this objective. So they developed two algorithms, first to capture whole arm events. Because we thought that from these previous studies, these is one particular type of event that are caused by some underlying mechanism. And then to look for who has complex rearrangements and have really objective estimates for that. And then this algorithm should be platform independent. And then they applied this to 3 big cohorts of a total of 595 patients analyzed by 3 different array CGH platforms. So first, to do the whole arm event classifier, that's what we call the Y. So this is a whole arm aberration index. So we use the normalized piecewise constant fitted value of the segmented algorithm or the segmented fragments. And then we would define that for each chromosome arm. And then we looked for the Y. At least 95% of all the probes on that arm should fulfill that criteria. And then we made a score to separate that a whole arm gain or a whole arm loss. So for each tumor, we scored each chromosome arm as gain or loss or nothing happened. So this is what you can see in these three cohorts, and now this is now the fraction, or no, this is now the Y score, and you can see for most of the chromosome it's centered around 0, so there really doesn't have any up and down, but for some chromosome it's clearly highly up here, and you can see that some chromosomes are really not affected by whole arm events, but some chromosomes indeed are, and this seems to be very similar for these three cohorts. So this is a 32K array on the oligo array. This is the Roma technology, and this is a 244K array on another data set. What we frequently see, you see the two chromosomes that are very frequently involved is 1Q and 16P. And of course, from old cytogenetics, we know that there could be some correlation between the 16Q and 1Q. So we set out to see, are similar Y on two chromosome arms across samples indicating our translocation? And indeed it is, for this case at least. So here we develop a FISH probe for each side of the centromere chromosome 1 and on 16. And you can see here from this tumor that you see here has a gain of chromosome 1Q and loss of 16Q. And you can see here that there are two normal chromosome 1, and it's a translocation chromosome, and it's a normal chromosome 16. So it appears like a loss of a whole arm, but it's actually a translocation. And we see this now that they translocate to 1, and they translocate to 8, and they translocate to 11. And then this was for the whole arm. The next was, of course, to look for these complex rearrangements, what we call the chi index. So here, for each breakpoint found by this piecewise constant fitting algorithm, 3 scores were calculated. First, the proximate to the neighboring breakpoints, magnitude of the change, and weight of importance. So the first step was to identify all the breakpoints and then the shifts in the segment and the type of the curve that this creates. Like, for example, this. So this is another type of curve, and this is another one. And this is step 1. Step 2 was to compute a score for the difference in amplitude between the segments, the neighboring segments. And then the step 3 was to compute the score as a function of the length of the adjacent segment. And then it was this last step was to calculate the proximity to the neighboring peak. So you had the magnitude of change and the weight of importance. And then you can get, this is the algorithm, and then you get chi-score for a chromosome arm, like for this sample, for example, and this is chromosome 7, which has a high chi-score, and this has a very low chi-score, and this has a very, very low chi-score. And you can see here, if you look, this is now not the distance, but the score to the next neighboring, and of course this is very distorted. If you have patterns like this, very little is happening when it comes to the chi aberration. And then they applied this to each chromosome arm, and each sample were calculated of this close to 595 breast cancer samples from 3 different cohorts on 3 different array platforms. And then first the Y identified 4 groups, which we call A to C. The first group was this group of tumors that had either gain of 1Q and or loss of 16Q. And you can see most of them have both. But some has only gain of 1Q, and a few has only gain of 16Q. And then they have some other events on top of that. Now the other group of tumors, which we call the B tumors, had neither these, and they had regional 5Q loss and 10P of 16, and they had this regional 5Q and 10P, 5Q loss and 10P gain. So this was a mixture of the tumors. And then we had a group of tumors that has neither of these events, which we call the C tumors. Interestingly, 3 of the 5 DCAS fall into this group. 13 of the 17 of the tumors with other histology fall into this group as well. So this was the grouping based on the Y, on the whole arm chromosome. So now you have all the 595 samples down here, and you have the chromosome arms on top here. Then we added the chi index. And then you can see that we can divide each of these groups again into two groups, low chi and high chi, low chi and high chi, low chi and high chi. And you can see that the chi are affecting different chromosomes, or different part of the chromosomes in different tumors, or different groups of the tumors, which are significant. So there are significant chi, for example, chromosome 11 never happened on the B and the A, B and the C, but it always happened on an A background of the Y group. So when we looked at the subtypes, looked at the centroid to the expression subtype, you clearly see that the A tumors, A1 and A2, are mostly of the luminal type. B, B2, and AB, and AB2, and AB1, AB both 2, and also some of the C have a basal-like phenotype when it comes to the expression. So now you really can further subgroup them using this type of algorithm. And this is the distribution. There you can see that they are distributed among the different subtypes. This is, I think it's interesting to look at, because this is a frequency plot that we very often see when you see this CGH pattern. And we tend to, oh, this is an 8Q amplification. But look, if you look at the 8Q amplification, what is when in the frequency, in the cohort like that, you can see that here is the frequency of what is caused by whole arm and what is caused by regional. And within chromosome 1 is almost all are caused by the whole arm. But on 8, there are only half of it, or a little bit more than half of it, which is caused by a whole arm. So the ones which are more local are of a different mechanism. And the same also for some of the other chromosomes. So these really say that looking at just some event on chromosome 8, you need to know why is it there and what type of event are there, not just the gain. And this has also survival implication. You can see if you have a high chi, you have a worse prognosis. If you have these are the 8 groups and they are really significant, whenever you add a high chi on top of the other Y groups, you get a worse prognosis. And this is also seen in the node-negative patients, and it's also seen in the untreated patient. This is independent of treatment as well. What I thought was interesting is to see if we look for grade. We know that grade is very much used in the clinic to stratify for the therapy. And here you can see that the grade doesn't have any influence in the B tumors and the AB tumors. So maybe you shouldn't use grade as a marker for treatment in the B and the AB tumors. And you can see here are some of the clinical B tumors versus A tumors, and these are in multivariate Cox regression, so it holds up and in addition to these. And treatment is also taken into the analysis here. So you can see this is this is for the chi, which is again give a twofold hazard risk of relapse. And this is now when you combine it, and here you can see that it really comes high up. So we think that high chi index is a poor prognosis in all the 4 Y groups. So it's independent of other prognostic factors. So how do they look like really? If you go now deeper in and try to do some deep sequencing. So we have in collaboration with the Sanger Center done paired end sequencing and selected out from these cohort and did paired end sequencing. And here you can see the type of this circle plot. So this means that it is a translocation between these two. What is interesting to see that this 116 is lost by the paired end sequencing. Because this is very close to the centromere. It actually is in the Notch gene, Notch2 gene itself, where it breaks and it fuse. And since the Notch are localized on both, you have Notch sequence on both sides of the centromere, we really don't know which side it breaks sometime. But that's missed by sequencing. But you can see here that they get more and more distorted and very different ones. So if you start to look more in detail, you can see that this is a basal tumor, very characteristic basal tumor by expression. These are now the circus plot, and these are the type of alterations you see by paired end sequencing. This is also a basal, but of the AB2 group. Expression-wise, they are very similar and has a very different CGH pattern and deep sequencing pattern. This has everything. The same is now for 2 luminal B, which belongs to 2 different classes of the Y-chi group, and the same is for ERB2. Also, they can see that this is very different from this one. So we set out that maybe this could be used to use this type of architectural distortion to see if this is a progression. So this is just a hypothesis that we think that the A1 to the A2 are a more linear type of progression. While if you go over to the B1, and particularly when you have some of these C2 tumors that are de novo, and they are all these strange type of the apocrine type, and are very different, because this has to be verified. But this is done on the CGH array, where we have not done SNP arrays. Because tumors, we know that tumors are very heterogeneous, and there are very many different clones with different percents of aberrant cells, and there are different ploidy, which we haven't taken into account in our previous algorithms. So these two postdocs, in collaboration again with Ole Christian, the statistician, set out to develop another algorithm based on the SNP arrays, which we called ASCAT, allele-specific copy number aberrations in tumor. And that was to detect copy number alterations, of course, as we did with the other ones, but to look for tumor ploidy and aberrant cell fraction and allele specificity, and more importantly, maybe allele skewness. Is it the skewness in losing one or the other allele? So are you predisposed to amplify an ERB2 of one particular allele, for example? So we know that there were algorithms out there to do non-aberrant cell infiltration scoring or to do aneuploid, and the ASCAT is trying actually to capture both. And that's what we think it does. You can look for gain and losses. You can look for copy number, of course. You can look for average allelic bias and look for LOH. Using the B allele frequency, we have used a 109K Illumina array here in this data set. We are now using a 660 Illumina and also the AFI on a much larger data set. And this is a goodness of fit to see what is the best ploidy that fit into these data that you can read off from the whole genome. And then you can calculate the probability of what is the ploidy, and we have counted some of the ploidy in many of these tumors, so we know that it fits. And also one important thing is that by using the SNP arrays, you can also look for copy-neutral events. And then you can start to see, apply this again to the same cohort of patients, and then you can start to see is this different between the different expression subgroups. And clearly you can see that the ERB2, for example, this is the percentage of aberrant cells. That means that this group of tumors has a lot of infiltrating other cells with no aberrant cells, which in a way we know that the ERB2 is the most inflammatory and catch a lot of them. What is maybe more interesting is to look at the ploidy. And if you look at the basal-like tumor, it has a peak which is below 2. So we think that and we also see from the next slide, we see that the basal-like has a very high fraction of LOH and copy-neutral events. So what we think is what happens in the basal-like tumors, or at least the B2 tumors, which we call them, which we think are the most typical ones, that they lose part of their genome, and then they start to amplify. So they go through a hyperploidy status before they hypo status before they sort of go up to 3, and then they go up to 6. You see the luminal are very well spread between the 2 and close to the 4. And this is the LOH. You can see the frequency of LOH. Again, you can see it's a huge higher fraction of LOH in the basal-like tumors. It may be that this is split into 2, but these are far too to say that these are 2 populations. And the same here for copy-neutral events, which we will not capture using the other arrays. Again, it might be 2 populations in the basal-like, but as I said, this needs to be verified. It was very interesting to see if there were some preferentially loss or gain in any of these using the SNPs throughout, or is it the preferential that are amplified or deleted. And you can see we do a huge number of comparisons. So this is now a Manhattan plot where you look for, and this is the p-values, and these are the genes that pass the Borresen-Dale correction, which actually are skewed towards amplified or deleted for one allele. And if you plot that, the allele skewness, so you can see that these red stripes are those that have passed the Borresen-Dale correction that are actually close to 100% one allele that is deleted or amplified. And there are some interesting candidates here, like, for example, the cadherin, and you have some of these strange ones up here. But because, again, this really needs to be verified, but we think now we have a tool to be able to do it, and we're doing it now on a much, much larger cohort. And now to apply the ASCAT algorithm first to correct for ploidy and infiltrating cells, and then apply the chi-y index on that again. So what are we trying to do? Well, we're trying to do methylation, microRNA, copy number, mRNA, proteins. We try to combine that. We haven't got there yet. We only have tools to combine 2 and 2, and then stratify for the third one. So we really would like to have more tools to look at that in a type of a structure. And of course, not to forget about the human genetic variation. This is all on a genetic background. So we know from previous studies that genotypes matter. BRCA1 carriers almost develop, all of them, basal-like tumors. And we know when we look at the SNPs, for example, in these which are captured by the GWAS studies, that they are very significantly differently distributed with the SNP and with their expression across these subtypes. So we think that it's very important to stratify for subgroups when you are searching for susceptibility genes when you do the GWAS studies. And of course, when we start now, this is some of the EQTL plots where we're looking for expression versus the CGA versus the SNPs, and you can try to look for correlation both in cis and in trans for which genes and which genotype regulates its own gene, which regulates other genes, and can be find network by looking at this. So this is one way for us to try to capture more than one level at a time. To end with where are we with our program? Well, we have this program now, which we have a consortium, which is called Oslo Breast Cancer Research Consortium, which I'm heading, where we really have our vision is to integrate molecular and epidemiological data. I haven't talked about epic data yet, so we'll just very, very briefly say a little bit about that in the very end. So, but this is what we're trying to capture, and I think that the metabolomics is very exciting because you can get this spectrum also in vivo on the patient itself. So if we can follow an expression profile that change or a CGH profile that change over treatment, we can maybe do that in vivo and just follow this metabolic spectrum to see what it captures. So my dream would be that a woman came in and you took an MRI or MRS, and then from the MRI and MRS you could tell, oh, this is a basal-like, and you start treatment, and then you start in a neoadjuvant setting, and you can capture the shrinkage again without any invasiveness, or take out the tumor at all. So for the time being, we have approximately 3,000 women with breast cancer and 3,000 controls, which we are working with at different levels. So you can see this is a group of women which are completely healthy, where we have biopsies from their breast, and we have blood samples and questionnaire data, which we are exploring. Then we have a group here which are enriched for DCIS. We have this population-based, and then we have this Oslo 1, which is our model cohort, which we have now almost at all levels. And then this Oslo 2, which we're aiming at 2,000. And this will also be done, the HR mass on almost all of them, and this will also be part of the deep sequencing in the International Cancer Genome Consortium, selected from that cohort. And we have other smaller cohorts. This is an older cohort that we have, which we have taken up again, and really can do some, because there's a lot of DNA there, so we can do a lot of these deep sequencing of that cohort, which has now more than 20 years follow-up, and this is another one with more than 20 years. And this is a neoadjuvant trial, which is ongoing, where it's only 35% recruited so far, but there will be multiple sampling during the time course of the treatment. This population-based cohort, there are 150,000 women in that cohort which we are following, and this cohort study is headed by Eilif Lund, who is the PI of that cohort. And it's been ongoing for many, many years. It started actually in 91 to do EPIC only, and then when I came on board, they said, oh, we need blood samples and we need to capture the tumor when it comes. So we were able to get funding to get out the questionnaires to 50,000 individuals born between 1943 and 57, and blood samples from 50,000 of them. And blood is drawn in these PAX tubes, as well as, so you have DNA, you have serum, and you have stored PAX tube where you can do the expression profiling of the blood cells. So these are 150,000 women which we are following. I usually say it's 150,001 because I'm one of them. And of course now we have registered data. These women go every second year to centralized mammograms, which are stored. And now within the next 5 years when this is started, we expect to have 1,500 breast cancer cases. We have 200 for the time being, and of course we lose some. But this is nationwide, and then we have questionnaire data. And every time a breast cancer case come in, 4 controls are called in and bled at the same time again, and a new EPIC profile is taken. And of course, at the time of tumor, we are doing blood sampling. Not all the sites do sentinel node and bone marrow, but those that come to our centers do. We take DNA, of course, and RNA, and we'll attempt to do all the analysis when we have this close to these 1,500 in the cohort. So I think at that time we can do all type of analysis to combine these, which is really population-based and where you can take in the EPIC data and take in the etiology as well. So where are we? So my vision is that, well, we are maybe a little bit above the sky. We have the patients and the tumors. We know something. We have good technology, which are improving as we speak, getting more and more high-tech. So we can get more and more done on smaller and smaller sample sets, or smaller and smaller samples. And then, of course, when we have the data from this, we hope to take it up and be able to do multidimensional analysis to find the pathway and the networks and the structures that can define the breast tumor in a particular patient, and model that and possibly find holes or Achilles heels that can be targeted both for prevention and for treatment. But I think prevention is also a big issue here because we have all these questionnaire data. And when we have that, of course, we have to take it into the in vitro and in vivo models to test that out, to validate and to confirm and to predict, and then take it back to the patients to translate the findings into clinical management. And then hopefully we will see the sunrise. So that's my vision. So this is my crew back home in Norway in front of our new research building. Highlight out the Ole Christian, who's the statistician, who's driving a lot of these young guys. And of course I have long collaboration, good mentor in David Bodstern still. The collaboration with the Cold Spring Harbor, with Uppsala, who we are generously giving us access to this DCIS biobank. And of course then the other international collaborators, including the Sanger Center. And then just put this up as a meeting, which I hope many of you hopefully maybe come to, which I think will be exciting if the volcano doesn't come up again, which will be in the summer in Oslo, as the European Association for Cancer Research. There are around 1,500 registered for the time being, which is still open. And this is not snow. This is the new Opera House in Oslo, where we will have the opening ceremony. So I welcome you there.
Time for 1 or 2 questions, if there are. No? If not, thank you.